In the last lab, we talked about DNA and replication and whatnot, and how it's so important that we replicate DNA when cells divide, but we didn't talk about the process of cells dividing. It turns out there's two different processes we're going to look at of how cells can make more of themselves: mitosis and meiosis. So let's look at a quick overview of both of these processes. Mitosis is a reproductive process where the parent cell creates two exact copies of itself. So we start with one cell and we create two identical cells, identical to each other and identical to the original parent cell. This is used for somatic or body cells. Cells like the ones that make up your muscle, that make up your bones, that make up your brain. These are all somatic cells. And since we're creating identical cells with two copies of each chromosome, we call them diploid daughter cells, or 2N. N representing the number of sets of chromosomes. So as diploid, di meaning two, we have 2N, two sets of chromosomes. We use mitosis for things like repair and growth. When you grow up, you don't make bigger cells, you just make more cells. And when you get injured, like if one of my many cats decides to scratch me, I need to replace those damaged and dead cells, and mitosis will allow for that replication process to happen and fill in the gaps where those cells were killed off. Meiosis is more particular. Meiosis is a reproductive process where the parent cell makes copies that are not identical. This is through a process called genetic recombination. And this is used for making sex cells, sperm and eggs. Because we're making cells that are going to be used for sexual reproduction, we don't want each cell to have both copies of chromosomes. We want the number of copies to be cut in half. We call this haploid, or N, where each cell has only one single set of chromosomes. The reason for this is because sperm and egg are meant to come together with the opposite, right? Sperm is meant to join with egg is meant to join with sperm. And if we had two sets each, we would have double the chromosomes we would actually need. By cutting the sets of chromosomes in half through meiosis, we can prevent extra chromosomes from joining in the mix. We also want to look at cellular division versus nuclear division. Cellular division, or cytokinesis, is where we're actually dividing the cell itself, the cytoplasm, the organelles, the plasma membrane, into two new cells. Whereas mitosis is the nuclear division. Mitosis is the phrase or the process we would call dividing the nucleus up, taking the replicated DNA and dividing it down the middle so that each new daughter cell has all of the genetic information that the original cell had. But it's not just DNA that we're dividing up, and we're using some other terminology when we're describing that. When we are looking at the DNA, we're also looking at some proteins that are associated with it called histones, and together the condensed DNA plus the histones is what we call chromatin. You'll see chroma and chromatin, chromatids, and centrosome, centromeres, and whatnot over and over and over in this chapter, so you'll really want to pay attention to the language here, as it can get confusing. So chromatin is condensed down DNA with histones. The sister chromatids are identical copies of DNA chromosomes. So before we go through mitosis, we have to make sure that the DNA has, in its entirety, been replicated. That way, each new cell gets one entire copy of every single chromosome. So the sister chromatids are the chromosomes that are completely 100% identical to each other. And so here we see a duplicated chromosome, that standard X-like shape we see here. And we see the sister chromatids. So one sister chromatid is here, and the other sister chromatid is over here if you're following my mouse. We don't have to worry about the P and Q arms or anything like that, but we do want to recognize what sister chromatids are, and we want to recognize where the sister chromatids are held together, called the centromere. I bring up the centromere and I emphasize centromere because there are other terms like centrosome that sound very similar. So centromere, centrosome, chromosome, chromatid, you can see how the language can get confusing. So on our chromosome, which is consisting of two chromatids that are held together at a centromere. But let's get into the process of mitosis itself. Like I mentioned before, this is where we are adding in new cells. Now, you're probably thinking, well, why not just make cells that we already have bigger? Well, cells have to be limited to a small size. It's important that their volume stays low in comparison to their surface area. This makes it easier for them to get the materials they need in, to move things around inside the cell, and to get waste products out. If the volume gets too high compared to the surface area by getting larger, then it can be very difficult for that cell to perform its functions. So since cells have to stay so small, multicellular organisms like ourselves have to make more cells in order to get bigger. When we're looking at these cells within multicellular organisms, we have specialized cells, muscle cells, skin cells, liver cells, so on and so forth. And each of these perform very specific functions and have specific shapes to them as well. We call that process differentiation, where these cells have differentiated into specific types of cells that will do specific jobs. But let's look at mitosis. How do we divide these cells up to make more when we need them? And there's a few different steps to this. So let's start before we even get into mitosis in general. Before we even get started, we need to replicate all of the DNA. Every single chromosome will get duplicated. It then starts to condense down in prophase. So these two pictures here are prophase. This is the early part of prophase and the later part of prophase. We start to then see that the DNA condenses down into that classic chromosome shape. The centrosomes, or the centrioles, are going to move to opposite sides to form what we call the poles. Think like a globe, like North Pole, South Pole. And they're going to start sending out little fibers that are going to grab on to our chromosomes at the centromere. This is prophase. We also see in this process that the nuclear envelope that was hiding this DNA away is going to disappear. It's hard to move chromosomes around if you have a membrane in the way, so we have to get that nuclear envelope out of the way first. So the nuclear envelope is breaking down, the chromosomes are condensed and visible, our centrosomes are becoming the poles and are sending out spindle fibers to reach our chromosomes. They've reached out, they've grabbed onto the chromosomes. Our next step is to line up all of the chromosomes along the equator, or the metaphase plate, the middle, so to speak, between the poles. And so it's like a tug-of-war game almost, where one pole is reaching out and grabbing onto the chromosomes on one side, and the other pole is reaching out and grabbing onto the chromosome from the other side. We line up in that middle. We call this metaphase. We then move to anaphase, and we see these two images here for anaphase. In anaphase, we are separating the duplicated chromosomes, the sister chromatids. We're separating them away from each other so that one sister chromatid will go in one way and the other sister chromatid goes the other way. By lining up these chromosomes and pulling the sister chromatids away from each other, we're ensuring that each new daughter cell will have one chromosome that is completely identical to the other chromosome from the other side, so that all of the chromosomes on this side up here are 100% identical to the chromosomes down here. That's how we conserve that genetic information. We don't lose any information and we don't gain anything extra. So we've pulled these sister chromatids apart, and one sister chromatid is moving towards one pole, the other sister chromatid moves towards the other. Once we've done that, we then can condense, or decondense I should say, back down to become what we looked like before, so we don't have that classic chromosome shape anymore, and the nuclear envelope starts to come back. We also see this pinching in right here called the cleavage furrow, which will help split our cell into two. We call these last phase telophase, where the nuclear envelope comes back, chromosomes become less visible and less distinct, and our poles and the fibers are going to start to kind of disappear. I like to think of telophase as the reverse of prophase. So if you remember what happens during prophase and you think about the opposite of that, that's probably what's happening in telophase. Once we've gone through that and we have two nuclei, we can finally split into two cells, and that's where cytokinesis can lead into that splitting process. So you'll want to get familiar with the basic description of each phase of mitosis. Prophase, metaphase, anaphase, and telophase. PMAT might be a little acronym that helps you remember that. Prophase, again, the chromatin condenses, we have visible chromosomes now. The centrosomes move to the poles. The fibers are going to attach to those centromeres. In metaphase, the chromosomes have been lined up at the equator. In anaphase, the sister chromatids are pulled apart from each other, going towards opposite poles. And in telophase, the chromosomes are going to decondense, become less distinct, nuclear envelope comes back, and the cleavage furrow forms, allowing cytokinesis to happen, and the cell can pinch off into two. But we're not always in mitosis. Mitosis is a small section within what we call the cell cycle. And we have to do many different things during the cell cycle to prepare for mitosis in the first place. So let's go through the basic cell cycle. We have G1, where the cellular contents are duplicated, just generic cellular contents. The S phase, which is where we replicate DNA, so I like to think of S as DNA synthesis. And the G2 phase, where we are going to double check and make sure that the DNA was replicated properly. There's no mistakes, nothing needs to be corrected, and then finally we can go into mitosis and cytokinesis. Collectively, we call G1, S, and G2 phases interphase. So if we talk about interphase, we're talking about all three of these together. But if you're specifically asked a question about when are cellular contents duplicated or when is DNA replicated, you'll want to know the specific letters. But mitosis is not the only process. We also have meiosis for when we want to make things like sex cells. As beings or organisms that use sexual reproduction, it's important that our gametes, the eggs and sperm, have half the number of chromosomes so that when the other half, the sperm or the egg, joins up, we reach that diploid state. Remember, diploid is talking about two sets of chromosomes versus haploid meaning one set. So a sperm or an egg would be haploid with one single set, and when the sperm and egg come together forming a zygote, we go back to being a diploid cell again. We also lose that identical nature we saw with mitosis. There's a recombination that can happen with those chromosomes, and we'll talk about how that can happen in just a second. But this is the basics of what's happening, and we can split meiosis into meiosis 1 and meiosis 2, but in general we see the same kinds of steps happening. Prophase 1, metaphase 1, anaphase 1, telophase 1, and then we go through the whole process again with prophase 2, metaphase 2, anaphase 2, and telophase 2. The general concepts of what are happening during prophase, metaphase, anaphase, telophase 1 and prophase, metaphase, anaphase, telophase 2 are basically the same. Regardless of which prophase you're talking about, the nuclear envelope will break down, the chromosomes will become visible. There is one small difference. I shouldn't say small, it's a pretty big deal. There is one difference from prophase 1 compared to prophase 2, and that's crossing over. We'll talk a little bit more about crossing over in just a second. But crossing over is a very important thing that only happens in prophase 1. In metaphase 1, we also have a small difference. Remember, we talked about metaphase being where we line up along the equator. And we do line up along the equator, but we line up along the equator slightly differently in metaphase 1 compared to metaphase 2. In metaphase 2, we line up single file and split the sister chromatids apart, similar to what we saw in mitosis. But in metaphase 1, instead of lining up single file, we line up in what we call homologous pairs. We'll talk a little bit more about homologous pairs and what they are in just a little bit. Same thing with anaphase 1, slightly different from anaphase 2, because since we lined up in pairs during metaphase 1, we are going to split according to those pairs, where one member of the pair goes one way and the other member of the pair goes the other way, as opposed to pulling sister chromatids apart. And telophase 1 versus telophase 2 is relatively unremarkable. They're about the same. So you'll want to remember the basics of prophase, metaphase, anaphase, telophase, since that will apply to any of the prophases that we, or metaphases, anaphases, telophases we look at. But you'll want to look out for the key differences in prophase, metaphase, and anaphase 1. They have a few key differences that you'll want to be aware of. So let's look at how we can increase genetic variation with meiosis, and this ties into what we were looking at with prophase, metaphase, and anaphase 1. The reason we're able to increase genetic variation is through what we call crossing over. Now this happens in that prophase 1 stage. Crossing over is where homologous chromosomes, or chromosomes that are the same number, so we have 46 chromosomes as humans in 23 pairs. So the homologous chromosomes are the specific pairs we're looking at. These paired chromosomes can trade pieces of DNA. And now it's important that we only trade with the correct chromosome. You don't want to trade away information with chromosome 3 if you are chromosome 7, right? You'd only want to trade information with the other chromosome 7 when crossing over happens. Think of it like when you were in school and maybe you had a cupcake in your lunchbox and a kid comes up to you and wants to trade his celery sticks for your cupcake. Well, that's not a very good trade. We don't want that. Instead, you'd probably be more willing to trade with someone who also has a cupcake that might be a different flavor. That's kind of the analogy we can use there. Because we've traded some pieces of DNA between the homologs, we no longer have this identical nature. The homologous chromosomes are paired together, but they're not identical the way sister chromatids are. They are very similar, but not 100% identical. So by trading pieces, we end up losing the identical nature of the sister chromatids. Independent assortment is also how we can increase genetic variation. With those homologous pairs, when they line up during metaphase 1, they're lining up together in those pairs. But who ends up on the left and who ends up on the right is random. There's nothing dictating which of the two chromosomes has to be left and right. And so you can end up with a lot of different combinations. For example, if you have 3 pairs of chromosomes, there's 8 different ways that you can line up in those pairs. If you look at the 23 pairs that humans have, we grow from 8 different combinations to over 8 million different ways to organize who is on left and who is on right in those pairs for all 23 pairs. That's a huge number. That's a lot of different ways that we can organize things, and that gives us the great variation we have. This is why you don't look identical to your siblings, because you're not identical to your siblings. There is a little bit of different information because of crossing over and independent assortment that you got from your parents compared to what they got from the same set of parents. So let's wrap this one up by comparing mitosis and meiosis one last time. Mitosis is used to create diploid, identical cells, and it's for growing and repairing tissues. we only use one cycle of division. So we go through the prophase through telophase process only one time. And we are not using crossing over or independent assortment since our goal is to stay identical with mitosis. Meiosis, though, creates non-identical cells with half the number of chromosomes. So for example, in humans who have 46 chromosomes in their body cells, the haploid sex cells would only have 23. And we do this because these are cells used for sexual reproduction. To cut the number of chromosomes in half, we are going through 2 cycles of division. We're going through that prophase through telophase process twice. And because we want non-identical cells, we are using crossing over and independent assortment to increase the variation so that all of the siblings and all of the children and offspring that we create with this sexual reproductive process do not end up identical to each other.